Good evening, everyone, and welcome to the Real Science Exchange, the pubcast where leading scientists and industry professionals meet over a few drinks to discuss the latest ideas and trends in animal nutrition. Now, tonight's going to be a little bit different. In this episode, we're going to be reviewing some presentations that were given at this year's ADSA. Now, the problem with the ADSA is there's such great content and presentations there that you just can't get to all of them. So with that in mind, we ask our technical team to scour the booklet and come up with some of the best content out there that we could share with you, our audience. And so that's what we're going to be doing in this episode. We're going to be going over 4 or 5 different presentations and papers that were presented at this year's ADSA for your enjoyment. So enjoy. Clay and I are here with a couple Spartans today. We've got Dr. Adam Locke from Michigan State and Jair Parales-Giron, who is a PhD candidate at Michigan State as well. We're going to be reviewing. Was it a presentation that you did, Jair?
I did both, but the abstract that we will be checking today is the oral presentation. Nevertheless, we can go through some results of that I present in the poster session if you want to as well.
So this was one of the spotlight poster presentations, correct? Where you did the 3?
No, I didn't do the 3-minute spotlight presentation. But I didn't do. But I present the poster I was part of the competition. I was in the semifinals for the poster competition, so that's why probably you are remembering the poster.
Excellent. Okay.
Nice. Now, the title of your poster was Whole Cottonseed and Fatty Acid Supplementation Impact Production Responses During the Immediate Postpartum in Multiparous Dairy Cows. And so why don't you set that up for us? What was the hypothesis behind the study and the methodology you used in the study as well?
Perfect. Thank you. And it's great to be with you guys again. This is the 3rd year in a row, so it's pretty cool feeling
you're
like
a pro.
Like a regular in the podcast. So our idea was revisiting some concepts because the latest or the newest report checking whole cottonseed supplementation in early lactation cows is from the 90s, and especially they were using whole cottonseed after the first 3 weeks of post-calving. So we are trying to supplement fatty acids to increase production responses and reduce negative energy balance, especially during the first 3 weeks post-calving. But also we want to check if there were any potential for using whole cottonseed during that period. We did some preliminary studies and some studies here checking different inclusion levels, both in mid-lactation cows. And our idea was giving continuity to that work, but also using oil seeds and, in our case, whole cottonseed. That's something that can be obtained and used readily here in the Midwest. So our intention and our plan was feeding whole cottonseed and fatty acids as well. So for that purpose, we use 52 multiparous cows in a randomized block design with a 2x2 factorial arrangement of treatments. So we have a control treatment without any supplementation, so no fat, no cottonseed. We have a treatment that includes 10% of cottonseed in the diet. Other treatment that was supplemented with fatty acids, 60-30 mix of palmitic and oleic at 1.5% of diet dry matter. And the last treatment was the combination of both, 10% of whole cottonseed and 1.5% of the fatty acid mix. So our idea was delivering the same amount of fatty acids coming from the whole cottonseed or coming from the fatty acid supplement. So we've been working with those mixes, and we have seen that those mixes could increase production responses, and especially the 60-30 could have some positive effects, also not only increasing milk production responses, but reducing body weight loss, but also we want to check if there's any potential for checking and including whole cottonseed during those periods, and that was our hypothesis. So having increases in production responses and probably obtaining an interaction between those two, improving production responses.
What were the differences in dietary fat levels between these 4 treatments?
The control diet was about 2%, and because we want to deliver like 1.5% coming from whole cottonseed or a fatty acid supplementation. So the whole cottonseed diet was about 3.5%, the fatty acid about the same content, and the combo diet, the combination was about 5%.
Just for my clarification, were those free fatty acids or in the form of triglycerides, and does it matter?
No, it was a calcium salt. It was a calcium salt of palmitic and oleic.
Got it. Yep.
So what were the results of this study?
We found some pretty cool results in terms of interactions between fatty acid supplementation and whole cottonseed supplementation. We see an increase feeding them individually, whole cottonseed or fatty acid, but in some meal variables, we didn't see a further improvement when fed both. So whole cottonseed increased energy corrected meal by almost 6 kilos compared to the control diet. The fatty acid supplementation, the energy corrected meal increase was around 5 kilos, but when we fed together, we didn't see any further improvement.
So how does the fatty acid profile of whole cottonseed compare to your fatty acid supplement?
Yeah, whole cottonseed is rich in linoleic acid, and I think that the second one is palmitic, and later goes the oleic. So our fat supplement was 60% palmitic acid and 30% oleic. So there are some differences in the fatty acid profile of the feed ingredients that we use to test our hypothesis.
Were you surprised by any of the results?
I am a little bit surprised about not seeing further improvement using both, but we've been discussing with Dr. Locke about it, and there are some logical explanations for not seeing further improvement. Nevertheless, we are still exploring and checking and doing and running some other analysis that could give us a better idea what could happen there. But we have some good explanations for tackling those results.
I think some of the interesting things are, if you look back, sort of like dogma or historical thoughts on this, is about don't feed fat to fresh cows. And I think Jair's done a series of studies now showing the potential benefits of specific fatty acids in that transition or early lactation cow. And related to that was one about lower fat diets or oil seeds maybe being a concern. But when Jair fed this was 10% whole cottonseed in the diet, we got 6.5%, 6.5 kilos of energy corrected milk over the control, and similar amount, about 5.5 to 6, I think, on with the 60-30 supplement, which we've done a lot of work with that 60-30 palmitic oleic. So there's some very good potential benefits there. Of course, one of the questions then is what happened to body weight? Did the cows lose more body weight? Remind me, Jair, that we didn't see any differences in body weight loss in this study.
And I have the answer for you. We didn't see any effects on body weight loss and body condition score loss.
What about dry matter intake?
Dry matter intake, that was something that was pretty interesting as well, because usually, and I always had like the idea, like whole cottonseed could be depressing dry matter intake in early lactation cows, and actually we observed a main effect of whole cottonseed increasing dry matter intake by 2 kilograms per day compared to the diets that weren't supplemented with whole cottonseed.
So a 6 kilo fat-corrected milk response and energy-corrected milk response. Small differences in dry matter intake. No difference in body weight change, where'd the milk come from?
We are still feeling more energy and more fatty acids coming from the cottonseed diet compared to the control diet. So that's part of the explanation.
I think that's the big part of the explanation.
Mhm.
I think the poster that Jair presented was on the digestibility results from this study.
Mhm.
His oral presentation was the production results. Um, you know, if you feed additional fatty acids in the diet that are highly digestible, such as these two sources, um, then you are going to get more nutrients into that cow, right, as well. So I think that's where we're getting the explanations there. There are some examples in the literature where you may feed a supplemental fatty acid in the diet, but not get more energy into the cow if they're not absorbed, or they have a very poor digestibility. So in this case, fatty acid absorption went up. Jair showed that in his post, which is probably one of the big reasons that we're seeing those benefits here as well.
Excellent.
Yeah.
Did you follow the cows? This is first 24 days in milk, correct? So did you follow the cows longer? Did the production differences continue?
Yeah, we check a carryover effect. We check carryover effect of treatments. I think our carryover period was like 3 weeks after ceasing feeding the treatments, but we didn't see carryover effects of the diets.
And that's actually different to other studies. So, it's
Yeah.
interesting to think through some of those differences. When we've looked at that 60-30 palmitic oleic blend before, we saw like 4 kilos of energy corrected milk carry through the next 7 weeks of lactation. And we've seen benefits again of it if we continued with that fatty acid supplementation. We got added on effects again. So trying to think through why we may not have seen carryover effects in this study, that's something we need to dig into more. But of course, we pulled back. If we'd carried on with these supplements or the cottonseed into that peak diet, I think we would definitely have continued to see production benefits.
Yeah, I think that's an important thing, Dr. Locke, because our carryover diet was saying this like fat free because we didn't have any whole cottonseed and any fat supplement during that carryover period and that carryover diet. Usually, our diets have some whole cottonseed, but we couldn't include whole cottonseed because it was one of our treatments. So we want to take that effect out from the equation. But yes, probably we have seen the 60-30 having carryover effects and probably having a carryover diet with a higher content of fatty acids, we could see that as well.
So for me, Clay, the biggest head scratcher still right now is why we didn't see an added benefit of having both, when we fed the cottonseed and the fatty acids.
Right.
We thought initially we'd get an added on benefit, but there's a number of things there to look at more, whether it's on the digestibility side, or whether one thought I have is we're maybe limiting on other nutrients to help continue to drive more milk fat. There's a whole host of interesting areas of research there, even around the amino acid side as well. What opportunities do we have to maybe try and work out? Could we have got more of a response? But right now, there's some good choices there, depending on what market you're in, what availability you have of different supplements or oil seeds. I think this work and other work we've done has helped to sort of revisit the whole oil seed area in general here as well.
Yeah. Excellent.
Well, Jair, yet another very interesting topic, and I want to thank you for joining us once again. Before you go, though, I understand that you'll be hopefully graduating with your PhD next summer, and so I'm just kind of curious what your plans are after that.
Okay, if everything goes well and if Dr. Doc doesn't have any new ideas for testing in the future, so that's the plan. But yes, I really like, I don't know, I am a passionate person about research, so probably doing research and development, but also I like work with the producers and visiting the producers and solve problems and doing troubleshooting at the farm level. So that's something that I really like. I am a passionate person about research and development, but also I love applied nutrition. So probably those two would be like the two areas that I am interested in continuing my professional life.
Well, I'm sure you're going to be successful, whatever it is you choose to do.
Thank you.
I appreciate you once again. And Adam, thank you for joining us as well.
Appreciate it. Thank you.
New research is changing everything we thought we knew about choline's impact on the cow and her calf, and top scientists have a lot to say about it. They are presenting new research that supports choline as a required nutrient to optimize milk production, choline as a required nutrient to support a healthy transition, choline as a required nutrient to improve calf health and growth, and choline as a required nutrient to increase colostrum quantity. This new research is solidifying choline's role as a required nutrient for essentially every cow, regardless of health status, milk production level, or body condition score. Learn more about the science that is changing the game and the choline source that is making it happen. Reassure precision-release choline from Balchem. Visit balchem.com/scientistsay to learn more.
And in this segment, we're here with Dr. Sabine Mann from Cornell University and Dr. Trent Westhoff from Cornell University as well. Now, Sabine, I'd like to start off. Why don't you just tell us a little bit about yourself?
Yeah, thank you for the invitation for today's recording of those ADSA presentations we gave. Always glad to talk about one of my favorite topics. I'm an associate professor here in the veterinary college at Cornell. I lead my own research program, but really the predominant time is spent doing clinical service to farms in the surrounding areas and teaching veterinary students, which is a great way of always knowing what the questions are out there. And that's one of the reasons why we're researching colostrum, because it's commonly something that is brought up on farms as a topic that folks want to know more about. It's an old topic. It's been around forever. We've talked about colostrum for a very long time, and still we find that there is more to learn. And Dr. Westhoff recently graduated PhD in my lab, took some of this research on and has developed a very keen interest in this topic as well. And I'm glad that he is able to join us today as well and talk about his research that contributes to the knowledge in this topic.
Yeah, perfect. Speaking of Trent, would you mind telling us just a little bit more about Trent and how he came to your lab and maybe some fun facts about Trent?
While I like to swim in lakes and don't want to know about the fish that are in there, Trent likes to fish in the lakes, and he continuously wants to tell me about how big they are, and I have to always stop him because I really don't want to know about the fish. How did Trent come to my lab? I reached out to a few of my colleagues in the field, including Dr. Lance Baumgart, who I have to credit with finding Trent. Trent was very adventurous in becoming my first PhD student. And we kind of developed the program in my lab together. It's fantastic to have a partner in crime to focus on what is relevant, what can we work on, what is applicable in the field. And he's been in my lab as a PhD student for 5 years and has recently graduated as a postdoc now, finishing up some of the work that we've done together. And then he will go on to do great things in the industry. And I look forward to watching him grow in that role as well.
Yeah, excellent. Yeah. So welcome, Trent. Trent, you gave an oral presentation at this year's ADSA, and that was titled Effects of Close-Up Metabolizable Protein Supply on Colostrum Yield, Composition, and IgG Concentration. Can you kind of give us just a kind of an overview of that trial? What was the hypothesis, protocol, etc.?
Yeah, thanks for the invitation to be here today, Scott. Super excited to be talking about colostrum, transition cow nutrition, as it's really two of my passions and something that I've really enjoyed learning about over my time in my PhD, and something that we continue to work on now. Yeah, so when we think about colostrum management programs or how much colostrum a cow gives, we know that there's variability associated in it throughout the times of the year, especially going into the fall and winter months in North America, and we commonly hear that as an issue this time of year. So we were really interested over the course of my PhD, and this probably started even before I arrived here at Cornell, of understanding what are some of those factors that influence colostrum production, primarily the yield and composition of the colostrum that those cows give. And we want to be able to create strategies to be able to implement or impact colostrum programs on farm. So over the course of my PhD, we started in an observational study where we worked with farms around the state of New York, and we looked at all these different factors: nutrition, management, individual cow factors, and we started to learn very quickly that there's so many different variables that are associated with colostrum production, and that's something that we knew from the literature, but we just provided additional evidence on multiple dairy farms in a different geographical location, and it's really started to expand our knowledge of different factors that influence colostrum production. But now to take that next step forward, I'm primarily interested in and very passionate about finding solutions that can be implemented on farm to make ground level impacts on farm. And that's kind of why we've focused more towards the management of farms as well as the nutrition. So in this specific abstract that we presented at ADSA, we were interested in looking at the effect of metabolizable protein supply fed during the close-up period. So we hypothesized that increasing the metabolizable protein supply of this study would alter the colostrum yield as well as the composition that those cows are given. So I'll just give us a brief study design. So we enrolled multiparous Holstein cows, 96 of them. And at 28 days before expected calving, we randomly assigned those cows to one of two prepartum diets. So those diets were formulated to supply 85 grams of metabolizable protein per kilogram of dry matter, or 39 grams per pound of dry matter, or an increased supply of metabolizable protein at 113 grams of metabolizable protein per kilogram dry matter, which would equal approximately 51 grams of metabolizable protein per pound. So we're also interested in removing the confounding effects of methionine and lysine supply, as we know the importance of methionine and lysine. There's tons of great literature. I think our nutritionists and our people in the field understand the importance of methionine and lysine throughout lactation, but definitely during this transition period. So we went ahead and removed those confounding effects by balancing both of those diets to supply equal amounts of methionine and lysine. And we formulated those at 1.24 and 3.84 grams of methionine and lysine per mcal of metabolizable energy.
Trent, before you go further, how do those numbers compare to what most people would do in the field or maybe look at as required for the lysine and methionine?
And just because we're halted, can you also a little bit talk about why we chose the levels of MP that we chose, just to give a bit of background for those that are listening?
For sure. So to answer your question, Jeff, those are probably a little bit higher than would typically be seen in lactating diets. But when we were going and designing these diets, there's not really good recommendations or requirements for a prepartum cow. So we went ahead and balanced our control diet, or the lower MP diet, at that 85 grams of MP per kilogram dry matter. And we did that to meet the Cornell, the CNCPS MP requirements during that close-up period. So we're at around that 100% of MP requirement. So we're not shorting these cows' metabolizable protein by means of our models. And then we went ahead and increased that to an elevated level, and that's going to be approximately 140% of model requirements, which is, thanks Sabine for asking that question. That's really good to help people understand those differences. between those two diets. And we did that with two main sources of protein: heat-treated soybean meal and a blood meal-based protein supplement. And then we took the high level of that high diet, and we just took the lower diet and added rumen protected methionine and lysine into those diets to match that of the high diet. So to directly answer your question, Jeff, that's probably a little bit higher than what we would typically see in lactating cow diets or those values that we're typically talking about. But in our diet formulation strategy, and because we don't have good recommendations at this point for that prepartum cow, that's how we went about the diet formulation.
Yeah, so when we tried to come up with useful levels of MP to test, we wanted to make sure we're not actually restricting these cows on protein, because we know that there is detrimental effects to that. So we knew that our control group was going to be approximately 100% of estimated requirements. And then we wanted to see if we elevated the supply of MP to, having a step that's significant to observe a biological effect here. Would we then see what we thought in our hypothesis was going to happen? So trying to make sure we weren't going to supply just 100 and 105 or 110%, which is maybe too small of a step to see those biological effects. And then if we were to see the outcomes we were hoping for, then we could work on titrating down, whereas potentially, that point where we would see the biological effects and dialing back a little bit on that MP, potentially. So that's how we came up with the original levels. There's kind of older research focusing not on MP, because that's a newer concept, right? So we really thought we needed to focus on MP and providing people with newer information on the relationship between colostrum synthesis and protein supply in a transition cow.
And I think that that decision, when we made that at the time, we had lots of conversations about trying to understand, we're currently recommending around that 1,200 grams per day based off of the CNCPS model as what we believe that requirement for that prepartum cow is. And it really depends on what your outcome of interest is. A lot of times we're evaluating based off of early lactation milk production, primarily during the first 3 weeks of lactation. So a larger scope of this study was to really try to refine some of these MP requirements or optimal levels, so to say, to feed during that transition period. Looking at colostrum obviously is the topic of interest for today's conversation, but the grand scope or the greater scope of this presentation was to really evaluate that cow throughout the entire transition period and see how we transition her into early lactation and then approaching peak lactation.
So now I've got some questions on that to what Sabine was referring to, but maybe before, can you just give us an overview of the results, what you determined?
Yeah, so in terms of colostrum production, those cows were fed for that 28 days before expected calving, and then within 8 hours of calving, we harvested colostrum. Those cows went through the parlor just as they would on a commercial dairy farm. We collected the weight, and then we collected samples for fat, protein, lactose, and IgG concentration. So we observed an interesting treatment by parity interaction for colostrum yield, such that those cows that were entering their second parity and fed the elevated metabolizable protein supply produced approximately 2 liters more colostrum compared to those cows that were fed that lower control level of MP. But when we look at those cows that are entering their third or greater lactation, we did not see a yield effect by increasing the metabolizable protein supply. So when we compare this to what is known, we know that cows in their second lactation produce more colostrum than those older cows. However, there's very little understanding how nutrition influences colostrum production in general, but very, very little information understood about how it impacts it in different parity groups. So we find that as a very interesting result of this study, but one that we would like to see additional findings or additional research be conducted to confirm whether that's true. But when we compare just the total colostrum yield across our sample population of animals, we're not seeing effect on colostrum yield. When it comes to composition and IgG concentration, metabolizable protein supply did not affect fat, protein, lactose, or IgG concentration.
Okay. Before I get to my real questions, I've got to ask, in your abstract, you measured IgG levels they were determined by radio immunodiffusion. So I've been out of the lab a while. Is that just a fancy name for Brix, or is that?
Yeah, so Brix is a measurement that we're using on farm. So radioimmunodiffusion is measuring the IgG molecules, which is that primary molecule that we're interested in for transferring passive immunity from that cow to the calf. Now, on the farm, it's not easily accessible, and it's very expensive to do things like radioimmunodiffusion to test the actual IgG concentration. So a Brix refractometer, which we're all used to using on farms, is an indirect assessment of the amount of solids, which is highly correlated with the IgG concentration in colostrum. So in addition to IgG concentration, which we do measure to understand the exact IgG concentration of colostrum, we also commonly use Brix percent in our research, as that is a more tangible number for those of us that come from farm backgrounds, work on farms, work with farms. And again, that was not affected. Brix percent was not affected by metabolizable protein supply in this study either.
So going back to the across the board, the lack of effects that you observed, I guess maybe that ultimate question, why do you think that is? What would you do different? Would you increase that spread? What are you thinking?
Yeah, so I think this is maybe a more complex problem than just in this individual study. So when we think about colostrum production, there's a lot of variability individually between individual cows. So in samples such as the one we're talking about, where we have just under 100 cows, when you have a lot of variability between individual animals, you need either a lot more animals to be able to detect a difference, or a smaller variability in the amount of colostrum that those cows are producing. So when we look across populations of animals across multiple different farms, we know that there's a lot of variability, and that variability is influenced by tons of different things: time of the year, parity, the management of those cows, the nutrition of those cows. So when we're trying to isolate individual effects, such as metabolizable protein supply, it can be quite challenging. So to statistically detect differences. So biologically, we can think about these numbers in the sense of how many liters of colostrum do we need to feed to a calf, and how do our numbers compare to that? So the treatment by parity interaction that I was discussing, we're talking about a 2-liter difference in colostrum yield, which I would consider a biologically relevant difference, as that produces approximately half of what you would feed to a calf at first feeding, or it could provide a second 2-liter meal to that calf. And that's part of the challenges when we go to interpret literature. We have to understand the sample size, but also the absolute values to understand whether those differences are biologically relevant towards the whole program. And with the goal of obviously providing calves with high quality colostrum, making sure that those animals are getting good, clean meals, and using those to start our calves off on the right foot.
Based on the Brix and the colostrum yields that you reported, I'm assuming this was done at the dairy at Cornell?
Correct.
The Brix and the overall colostrum yield looks pretty good all around, at least compared to what I see out west more so. And maybe I know part of that's seasonal, but do you think even those control cows having such good quality and quantity of colostrum may have affected? Would we see more of a result if that Brix level was lower to begin with? Because was it like 26 and versus 27? I see a lot of 22s, 23s in the field.
Correct. Yeah, I think that's something that we notice typically with working on the herd that we do a lot of our research on, is the colostrum production is typically quite good. So we ran the study throughout the summer months, which obviously we're gaining a bit of yield. We're at the seasonal peak of our yield when we ran this study, but there's potential for that. I think when we think about the design of the study and the exact question that we're asking probably a bit of a different question. This goes back to what Sabine brought up earlier, where we were not limiting these cows on metabolizable protein. We were trying to understand, by providing additional protein, is their effect on colostrum production. Our results, and when we compare the results to the literature, would suggest that metabolizable protein supply has quite little impact on colostrum production to our knowledge currently. But we're not asking if we increase it to a point where that cow is not limited so much, does it affect colostrum production? We're asking if we provide that animal more MP than what we think is required at this time, if that'll affect her colostrum production. Okay. But yeah, overall, I think our colostrum production was quite good in this study. And in terms of providing high quality colostrum and enough of it for a feeding program, we did accomplish that in both of our treatments.
And to your question, why do you see, colostrum being different in different regions of the country or within a region between different dairies? Trent's done a nice job reviewing the available literature on this and put that together in a review. And I know that's not part of the abstract you presented, but Trent, do you want to just briefly summarize the different factors that we consider as influential on colostrum production? As far as we know, this is only what we think we know. And there likely will be more as we do more research that tell us, can we manipulate certain aspects on farm and close the gap for farms that produce really good quality colostrum and those that may have opportunity to do so.
Yeah, so in our latest review that we wrote, we kind of broke it down into 3 different factors that influence colostrum production and quality. The 2 of those factors come before the time of calving, which would be cow factors, and then management and nutrition factors. And the third one we include in that is post-harvest management. So when we talk about cow factors, there's many of them, but parity, time of the year when that cow is going to calve, the temperature and humidity that cow is exposed to, which kind of fits into management. On the management and nutrition side, we talk about different nutritional components. So we're talking about MP here today, starch level or metabolizable energy level, how long that cow is in close-up pens. Depending on your study and the location that study was performed in, you get a number of associations. So obviously when we think about associations, we have to consider that by manipulating that variable prepartum, that might not give us a consistent effect, as it's an association rather than a causation. But then within that third bucket of post-harvest management, we talk about how quickly that colostrum is harvested, how quickly we cool it to preserve, or how we store it, refrigerator, freezer, or does it go directly to the calf? And when we think about all these pieces that come together, that in total manipulates the amount of colostrum that is harvested and the quality of colostrum that is fed to that calf. And I think that goes back to your question, Jeff, of why we see that in different geographical areas, is because a lot of these variables that we're talking about are affected by location. So if we go to the Southwest, obviously the environment and the management style of those dairies are going to be completely different than we're experiencing here in the Northeast or the Midwest, or if we go to the West. And all of these things we think come into affect colostrum production and the quality of colostrum, which is why it makes it so challenging for us to just give this silver bullet answer of how we can fix that problem, is because we know that there's handfuls, up to 20 or so variables probably that influence it. And every individual farm, even if they're just down the road from each other, could be struggling at the same time from two completely different things, which is makes consulting that issue so challenging.
So is that paper, is it in process? Has it been published?
Yes, it's the invited review that we are discussing right now is published in the Journal of Dairy Science, and the effect of metabolizable protein is also published in the Journal of Dairy Science. But that invited review goes through a lot of different factors, and I think does a pretty good job of highlighting variability and different factors to consider. And we even have a little figure at the end of that paper that kind of pulls everything together for a quick look.
Great.
And those are accessible online for free. So anybody can look those up if they want to.
We could even provide a link, I believe, in our show notes if that would be appropriate.
Yeah.
Well, Trent, when Dr. Sabine introduced you, she said she's looking for some great things from you in the future. Why don't you tell us a little bit about what the near-term future looks like for Dr. Trent?
Yeah, so finished up my PhD a couple months ago, and in a short postdoc, working in the lab, working on finishing up some of the data for my PhD, as well as we have a couple other projects that we've got excited about over the last couple of years and have started. So working on keeping those going and hopefully completing some of those before my short postdoc finishes. And then I'm looking for my next industry opportunity as we speak.
Going into industry. Very well.
Correct.
Love to see that resume when you have when the time comes. All right. Very well.
Glad to share.
Yeah. All right. Thank you both very much for joining us today. This has been very interesting. We appreciate you.
Great.
Thanks
Thank
for
you
having
for
us.
the invitation.
Chatting.
You're welcome. Balchem has invested decades perfecting the art of nutrient encapsulation. And now, new AminoShure XL is setting a new standard, delivering 35% more metabolizable lysine than leading competitors. With AminoShure XL, you can provide your cows with a more consistent and higher quality source of metabolizable lysine, leading to improved and more dependable results. A recent meta-analysis found that feeding rumen-protected lysine led to an increase in milk fat percentage and 4.4 more pounds of energy-corrected milk. Feed new AminoShure XL to your cows and have confidence that you're using the most consistent, reliable, and cost-effective source of lysine on the market today. Learn more at balchem.com/XL. And our next guest is once again Dr. Sabine Mann from Cornell University. Now, Sabine, you gave a presentation at the ADSA this year. It was at a symposia called the Colostrum, the role it plays in calf health, development, and future productivity. And then the title of your talk within that symposia was titled Colostrum, More Than Just IgG, Colostrum Components and Its Effects on Calf Health. So why don't you just start off by giving us kind of an overview of what that talk was about?
Yeah, thank you for letting me speak on this topic. As I mentioned before, it's one of my favorite topics. I wanted to focus the talk at ADSA on giving credit to the importance of IgG, but also extending our view on all the other components of colostrum. We traditionally have focused on the immunoglobulin component, and among the immunoglobulins, IgG, one of the subtypes, is really important. We've known since the beginnings of the 1900s that calves are not able to survive if they don't ingest enough IgG. But what I think we sometimes forget is all the other components of colostrum are not given in isolation from IgG, right? So as we give the calf IgG, it also gets the benefit of bioactive factors in colostrum, nutrients in colostrum. And I wanted to focus this talk just reminding ourselves of the importance of these specific components.
All right, and then how would you quantify, these nutrients and biofactors, their importance relative to IgG? Is it, just as important, or how would you quantify that?
Yeah, so that's a tricky question to answer because I think we're still learning more about the role the bioactive factors have in the development of the calf and its future productivity and health. For example, we've learned a lot about insulin, for example, or IGF in the maturation of the gut and the role that these two bioactive factors likely have in the success of the calf. How can we quantify that when we have almost 100 years of research in IgG, but we're only now dabbling into the other components? I would say that it is hard to say, they're equally important or more important or less important until we have research where we would give these components in isolation and study the effects. And that's very academic, right? Nobody will take out IgG and give it by itself, and somebody will then take out insulin and give it by itself. So that's probably going to be something that in the near future we can't answer. My gut feeling, however, is that all of these components have an importance, that there is a reason why they're in colostrum in the amounts that they're in there, and that we just have to get more data, especially field-applied data, data from calves in larger numbers to figure out what the potential biological effects could be. And those studies have been done to some extent, but mostly in smaller cohorts where we add specific components to colostrum and then try to isolate the effect of that specific component. As you can imagine, that's a lot of involvement, a lot of cost in some of these components to try to add them. So it's not as easily done as IgG. I do want to remind the listeners, though, that we use IgG as a biomarker of good colostrum management, right? So we give colostrum and we, in the end, after everything has happened, the calf's been fed, we measure IgG in blood, and we then assign the health effects of colostrum to IgG. However, there's a potential that when we measure this IgG, we're also only using it as a marker for all the other components that have been transferred as well, right? We're just not looking for them as we're looking for the IgG. And Jeff, you've mentioned the Brix refractometer before. There are some fantastic estimations of IgG in the field that are very cost effective, and you can do them calf side. There's very few bioactive factors you can measure as easily as you can estimate or measure IgG, right? So that's hampering our understanding of the importance as well. That was a very roundabout way of saying I think they're all important, but don't ask me which numbers, in relation to each other.
Yeah.
So you mentioned, I want to get in the weeds just for a second before we get to more of the management things, but some of these compounds I've at least heard of, cytokines and peptides, et cetera, but you also mentioned complement.
Mm-hmm.
What is that? Or what does that mean?
Yeah, complement's a way to scare every undergraduate or veterinary student into, having to remember all of these components. Some of us may have some PTSD having to remember all the complement components. If you have had some of this biology in your training before, you may remember there's a few different factors that come together and they make a whole into a cell, and then by doing so, they kill that particular cell. That's the job of complement, among others. And complement is just a way of the organism to perform its innate immune functions, meaning that it's faced with a pathogen, it's faced with a threat, it can immediately react to this pathogen much, much faster than an organism building immunity by producing immunoglobulins, for example, right? So complement is there, it gets activated a few different ways, and it kills pathogens, it kills bacteria. That's the most important job it has.
Okay, gotcha. So I thought, and I listened to your talk, and I thought the interesting part was about, from a management standpoint, where we heat treat colostrum on farm. And you brought up the point that when we're heat treating to remove, bacterial contamination, are we potentially harming or maybe degrading some of these beneficial compounds? Can you expand on that a little bit, and what do we do with that?
Right. So as you said, part of what I wanted to bring up in the conversation was we do certain post-harvest processes, including heat treatment. Heat treatment has a place in controlling bacterial contamination. Heat treatment has a place in reducing pathogens that we know are potentially transmitted in colostrum. But when we have assessed the effects of heat treatment, we've always focused on IgG concentration. IgG is a protein that's very heat stable, or fairly heat stable at least, at the concentrations that we find in colostrum and at the temperatures and times that we expose colostrum to heat treatment for. But the other components of colostrum, including, for example, immune cells, complement, insulin, IGF, even IgA, which is another immunoglobulin that's providing mucosal defense, are all less heat stable than IgG. And we could show in our research that these components are either reduced in their activity or completely obliterated, such as immune cells are dead at the end of a heat treatment process. Complement is much less effective at the end of a heat treatment process. We lose some insulin and IGF-1 concentration. And all of these together may change the way that colostrum exerts its biological function in the calf. Again, we have a lot of research on IgG, many, many years of research, but we have much less research on the impacts of these other components and how, when we change their function and their concentration by heat treatment, we may change the biological effects in calves.
Yeah. So yeah, my first thought was, let's assume that we are changing those biological effects of these other compounds by heat treatment. What does that mean on farm? Is it going to be less heat treatment of colostrum? So we're going to have to get really, really good at controlling contamination by other management means. But
Right.
That would be a huge barrier, trying to convince figuring that out and getting better than what we are.
Right. And, I go to lots of farms, and it's a very hard thing to do. Harvesting colostrum in a clean fashion is not easy, as we know. And then we store it, right? And we have to rely on the bacterial numbers to be low for the storage. And so heat treatment helps us. It's a little bit of a band-aid for some of the management processes that we have. And I would not suggest we stop heat treating. I think what I'm suggesting is we need to be aware of potential effects we're having from this heat treatment process. And if these are of significance, if they turn out to be of significance, maybe we need to figure out either ways of then adding these components back into colostrum, if we can, finding other ways of getting these biological functions, or giving people the information of, you get the benefit of heat treatment, but you also get a detriment. So you have a choice then to pick a route of your colostrum management on farm. And if you don't need to heat treat because maybe you're excellent in your colostrum harvest and you don't need to control pathogens on farm, maybe then for those producers, it's a better way to just not go through the heat treatment process. I think we don't have the data yet for me to, be here and say, let's not heat treat, because there is lots of benefits to heat treatment, and all of us are aware of those. But I think we have more to learn about the other effects of heat treatment.
Well, and it might just be fine-tuning our heat treatment processes, because been on a lot of farms where colostrum quality is good, but there's some mortality and morbidity, and it's because of the heat, maybe overheating.
Yeah, this is an excellent point you bring up. The IgG molecule I said before, is quite heat stable, but just a few degrees centigrade difference in the heat treatment process will render this protein inactive. So you have to really make sure you're tuned in with the heat treatment equipment. And I recommend that once in a while you check the temperatures in your water bath, or you check the temperatures in your equipment. Maybe get the help of the company that provided this equipment to you, and make sure that you really are doing the right thing with your colostrum, because, as I said, just a few degrees difference might actually inactivate IgG to a large extent.
So I've got to ask, because I've ran across this recently, what is a few degrees? Is it 2 degrees, 3 degrees?
Yeah, so our recommendations based on good research, solid research, are 60 minutes at 60 degrees Celsius, right? We know that the IgG molecule is starting to degrade at 63 degrees Celsius.
Okay.
But you also have to keep in mind, we usually treat a bigger amount of colostrum either in a bag or in a batch. And sometimes we have problems with that colostrum not evenly being exposed to the heat. And if you have heat exposure in some parts of the colostrum and not in others, then you're undertreating parts of it and you're overheating others. So it's very important that you get good agitation of the water bath, you get good mixture in a batch pasteurizer, so that you prevent certain subparts of that colostrum being under or overexposed. Okay. That makes sense.
So, Scott, I could ask a 10 more questions, but I've just got one more, then we'll let you take back over. So, Sabine, as far as, we're measuring IgG via bricks on farm, and, technologies keep improving. And, a few years ago, we figured out how to test BHBA for, for transition cows. So that technology is going to improve. So what would be the next component do you think we might be able to measure? Or maybe the better question is, which one would you like to be able to measure from colostrum?
Yeah, that's an excellent question. So it's important that we know how much IgG is in colostrum. No doubt about it. And so if I had wish, I would like to assess the antimicrobial activity of colostrum. I think if I had a way of, on the farm being able to say the antimicrobial activity of this colostrum is with all the components that are in there is X, that would be fantastic. Another thing, because I can never only, have one wish, right? Another thing that I would want to know is what is the insulin IGF component, so the growth factors, right? We are still working on an understanding of, can a cow have colostrum with high IgG but low growth factors, or is good quality colostrum measured by IgG usually an indicator that she would have all these other components as well? We don't have a lot of research on that yet. So I think it would improve our understanding of judging good quality colostrum versus poor quality colostrum, extending that beyond just IgG.
Okay, great.
Yeah, great questions, Jeff. Dr. Mann, your presentation was part of a larger symposium. I'm kind of curious, were there any key takeaways or any big ahas that you came away with from listening to some of the other presenters?
The biggest takeaways are usually the great questions we get from the audience, right? Reminding us where we really need to put our focus. I think that one key takeaway is that we're working on some of these nitty-gritty ingredients, and maybe we still have to work a little bit more on understanding what holds people back in on-farm excellent colostrum management. We've known what excellent colostrum management can look like for a long time, but I think we need to focus maybe, and this is a good reminder for myself, on providing easy ways for producers to implement the things that we put forward in research, right? So what are easy ways for somebody to harvest very clean colostrum? What does that mean? Can we find some more recommendations? Can we provide some more data on how is the cool down process done the best way? What are the storage lengths that we suggest? Are there other additives than the ones we're aware of right now that could help folks extend the shelf life of their colostrum? A simple question is, should every calf be fed a gallon of colostrum? I think we still have some things to focus on that the audience reminded us in that nice symposium, where a lot of us were focusing, as I said, on kind of the smaller molecules, etc., that are not inherently useful for somebody making decisions on farm, but all contribute to this understanding of the importance of colostrum.
Yeah, great input. And then just kind of in closing, is there any kind of key takeaways that you'd like to leave with our audience from your presentation specifically?
Yeah, I think I would remind the listeners that IgG is important, but for a long time we have measured IgG transfer as the sole way of establishing good colostrum management, and want to really leave folks with the thought, what if IgG is also a marker for all the other things that are in colostrum, and we've just used it thinking that IgG maybe is the one important component. And maybe a second thought is that we can focus a lot on making the best quality colostrum, changing how we manage transition cows, changing how we harvest it, but we still need to get it into the calf, right? We need to get it into the calf. And sometimes when we see breakdowns of colostrum management on farm, it's not that we don't have good quality colostrum, but that somehow we have a breakdown in getting it into the calves in a good, clean, safe fashion, in an adequate amount, and at a good temperature. And this is a good reminder for us who live in colder climates in the winter. We have to make sure colostrum is an adequate temperature, so we don't provide a bunch of cold colostrum to a calf that makes it harder for it to take up the good nutrients and the good bioactive factors and IgG from that colostrum.
Dr. Mann, this has been great information, and I want to thank you for joining us here at the Real Science Exchange. It's much appreciated.
Thank you very much for the invitation.
You're very welcome.
Thanks.
And to our loyal listeners, thank you as well for joining us here once again at the Real Science Exchange. We hope you learned something. We hope you had some fun. And we hope to see you next time here at the Real Science Exchange, where it's always happy hour and you're always among friends.
We'd love to hear your comments or ideas for topics and guests. So please reach out via email to anh.marketing@balchem.com with any suggestions, and we'll work hard to add them to the schedule. Don't forget to leave a 5-star rating on your way out. You can request your Real Science Exchange t-shirt in just a few easy steps. Just like or subscribe to the Real Science Exchange and send us a screenshot along with your address and t-shirt size to anh.marketing@balchem.com. Balchem's Real Science Lecture Series of webinars continues with ruminant-focused topics on the first Tuesday of every month, monogastric-focused topics on the second Tuesday of each month, and quarterly topics for the companion animal segment. Visit balchem.com/realscience to see the latest schedule and to register for upcoming webinars.